So a few days ago, I was trying to be a good husband and build a new shed for my wife's horses. Naturally, being the technically skillful individual I am with tools, I got a boo-boo, and now it looks like it's starting to get infected, see? So now I'm going to die. Oh wait, no. I have an immune system. An innate immune system. So now the innate immune system is a part of the more immediate reaction to infection, but generally takes a little bit more time and does require some help from the complement system in order to get things going. The three main components of the innate immune system are the macrophages, which we've already met briefly, the cytokine system, and the neutrophils. Now with macrophages, which we've already talked about, are the garbage men of the immune system. They run around picking up just about anything. They have on them a variety of receptors that are able to bind to pathogens from a wide variety of sources, and from that binding is able to trigger phagocytosis. As we talked about last time, C3b is a good binding substrate for phagocytosis for macrophages, but there are a variety of other receptors on the macrophage that it can use as binding substrate. Lectins, lipoproteins, LPS of gram-negative bacilli, tectonic acid that's present on gram-positive cocci, and a wide variety of other bacterial proteins and complement can induce phagocytosis in a macrophage. Unlike a lot of antibody-antigen interactions, once a binding occurs on a macrophage, this binding is irreversible, and then it induces an endocytosis. So once you're stuck on a macrophage, you're stuck with it for the rest of your now very short life. Once you've been completely endocytosed, you've become part of what is called a phagosome, and this phagosome then becomes complexed with lysosomes, which contain a variety of different nasty chemicals to help destroy you. Macrophages also have the ability to detect the presence of lipopolysaccharide, or LPS, and when it detects that, it binds it specifically to a toll-like receptor 4 and CD14 on its cell surface, and in that binding initiates a series of intracellular cascades to activate transcription of nuclear factor kappa B, which for those of you who are more experienced in critical care will understand is part of the inflammatory cascades. Nuclear factor kappa B then induces transcription of cytokines, adhesion molecules, and a wide variety of other inflammatory mediators to really ramp up the body's innate and induced immune system in response to this pathogen. Cytokines are the next cog in the wheel of the immune system. Cytokines are small proteins and that are used primarily as a communication method in close between cells. Binding of a cytokine induces some form of an intracellular signal on a wide variety of different cells that can alter that cell's specific behavior. One of the things that's important to understand about cytokines is that a specific cytokine, when it binds to a variety of different cell types, can have differential effects. So where a cytokine can induce inflammation on one cell type, it can induce coagulation or the release of other cytokines or other inflammatory mediators on a different cell type. Cytokines are small molecules and they are only short-living, and so can only really work within the local environment, and so are very useful for coordinating the battle and on the battlefield, but are not as well designed to go out into the world and broadly communicate a signal. Once a macrophage becomes activated, it can secrete a wide variety of cytokines that alert the rest of the system to the presence of an infection. Some of these include IL-1 beta, IL-6, IL-12, and TNF-alpha. All of these cytokines are considered pro-inflammatory cytokines because they actually ramp up the state of inflammation both locally and then also throughout the rest of the body. The inflammation not only recruits cells such as neutrophils, to the site of the infection, but also creates a more of a conducive working environment for neutrophils and give them access to the site. So part of the reason why when somebody develops inflammation, their cells become leaky is to actually permit the neutrophils to get into the source of the infection so that they can start doing their job. Now, neutrophils do most of the heavy lifting within the innate immune system. These are, in a sense, very specialized cells that, unlike macrophages, which are more general garbage men, are actually dedicated killers that circulate through the blood, waiting for the call to duty, and then when they get to the site, they get to work. Macrophages, as they have a role in releasing cytokines and inducing inflammation, help recruit neutrophils to the site of infection to get into the battle. In many respects, neutrophils are similar to macrophages in that they phagocyte a variety of different pathogens relatively non-specifically, just like macrophages do. And they also carry many of the same proteolytic and phagocytic receptors on its surface membrane in order to respond to complement coatings on the pathogens in order to induce phagocytosis. However, they carry many more different antimicrobial weapons in their armamentarium, and critically, they are actually programmed to die young. Once a neutrophil engulfs a bacteria, it has 3 different sets of granules that it fuses with the phagosome in order to kill this organism. While the details are not necessarily very useful in day-to-day operations, they are actually really neat in the way in which things go down inside the neutrophil. First, there's the production of hydrogen peroxide inside the phagosome where the pathogen has been captured in. This hydrogen peroxide not only has its local effects on the pathogen itself, but it also raises the pH of the phagosome and allows it to activate all of the enzymes that are present in the phagosome, which the neutrophil keeps as an inactive form because they're so highly dangerous and volatile that it would induce death within the neutrophil itself if they were present in the active form. So the presence of the hydrogen peroxide allows these enzymes to become activated and then get to work and start doing their job. The next step after the activation of all of these enzymes is what's called the respiratory burst that produces a large number of toxic oxygen species which do the pathogen in once and for all. Unfortunately, these toxic oxygen species can escape from the neutrophil and can go on to cause collateral damage at the site of the infection, accelerating the process of inflammation locally. Now, unlike macrophages, which are very long-living organisms, the neutrophils are actually programmed to die young, and they can't reload, so they can't reform these granules once they've been produced, once they've been consumed. There's nothing else that neutrophil can do, and so it goes on to die from a process called apoptosis. The macrophages then come by to clean up the remnants of their fallen comrades, and any residual pathogen that may have survived all of this is then finished off inside the macrophage. So that's the innate immune system, and today we learned about 3 components of the innate immune system that are induced after the development of complement and activation of the complement cascade. We learned about the role of macrophages not only as a cleanup crew in a non-specific phagocytosis, but also in the production of cytokines, which are small molecules which facilitate intracellular communication, and then the role of the neutrophils to do the final job of cleaning up a lot of the infection, where they're different than macrophages in that they can't live for long. Once they phagocytose something, they have to die. Coming up, we're going to talk more about the cellular components of the immune system and start focusing more on the humoral as well as the lymphocyte portion of the immune system.